Good morning, good afternoon, good evening from wherever you may be watching. My names are Bernice Waweru, a bioinformatician at the Becker Ilri Hub at the International Livestock Research Organization here in Nairobi, Kenya. I have a background in plant breeding and biotechnology through a master's degree at the University of Eldoret and bioinformatics training from an initiative that I will be talking about today. I want to present to you about orphan crop genomics 2.0, developing capacity for genomic research in Africa. I want to start by taking your thoughts back to the Green Revolution. I myself began my research career as a young scientist working on wheat. Dr. Norman Borlaug remains a great inspiration. He's among the key figures prominent in averting mass starvation after World War II through introduction of key technologies. The father of the Green Revolution helped traditionally food-deficient countries become self-sufficient in relatively short times, saving hundreds of millions of people in India, Pakistan, Syria, Tunisia, and North African countries like Algeria. One of Borlaug's greatest legacies was in training the next generation of scientists to sustainably continue and scale the technologies developed through capacity building, evidently seen in this image. I myself, I am a recipient of the Borlaug Leap Award, inspired by his love for teaching and training. Consequently, these countries in the Middle East and North Africa are among the key producers of staple crops consumed today in the world. Africa, however, seemed to have been left out, and we are yet to catch up, but all is not lost. Africa, I want to point out, has a rapidly growing population. It is estimated that 1 in every 4 people living in the world in the next 3 decades is going to be from Africa. Estimates on production based on current yield gaps, however, suggest that we might not have caught up with our production to sustainably feed the growing population. To sustainably achieve a balanced and sustainable food security, we need to diversify our food sources to include underutilized crops that can help to increase diversification of food production and to add new species to our diet that can help a better supply of nutrients, including amino acids, fiber, and proteins. With a diversity of 370 or 369,000 known plant species, only 7,000 of these were used as food by early human societies. Currently, only about 250 of those plant species are cultivated. And yet, only 3 of those are the major staple crops grown in the world, that is maize, wheat, and rice, that account for 50% of the global calorie production and that calorie that is consumed in the world. To achieve a balanced and sustainable food security, we must diversify our food sources. With this slide, I want to demonstrate a few things. One, we already have quite a diverse source of calories in Sub-Saharan Africa being produced, although we are still importing quite a high amount of wheat. We also produce quite a significant amount of orphan crops within Africa, despite the fact that most of these are grown at a subsistence level, they still flourish. I also want to point out that from these traditional crops, we are able to get quite a high level of nutrients. They provide majority of the calories consumed in Africa, 29%, as well as majority of the protein that is consumed, 34%. So what we need to do is to rather take advantage of the already existing resources and scale them up to meet the demand required to feed our continent and eventually feed the world. Thankfully, low and middle income countries, including those countries in Sub-Saharan Africa, have a rich variety of these underutilized crops, which will be the focus of my talk today. These crops are sometimes called orphan crops or even sometimes forgotten crops. Although very nutritious, most underutilized crops are underdeveloped and underused because they have quite a number of production constraints, including long growth periods, long processing times, and sometimes they also produce toxins. Genomics is, however, providing a way to develop these crops for food security in sub-Saharan countries and other low and middle income countries. I would like to demonstrate this with an example. This is grass pea. It's a nutritious and climate resilient crop that can survive under extreme drought. It, however, causes a disease called letharism when eaten in high quantities because it produces a toxin. However, thanks to the advent of genomics, scientists have now identified genes that can be used through genome editing to reduce the amount of toxins that are produced by grass pea. Despite or besides the benefits in nutrition, orphan crops also provide benefits in economies. Using genomics to accelerate the development of orphan crops, these countries can reduce their over-reliance on importation and reduce the impact of importation on their countries. Low-cost portable sequencing technologies are changing the way we do research. It is now known that sequencing is becoming quite cheap, and there are various sequencing platforms that are available, and some are more suited for low and middle income countries, including those in sub-Saharan Africa, than others. All of them have their pros and cons. Today, however, I want to focus on sequencing using the Oxford Nanopore MinION that produces third generation long reads. Technologies like the MinION are opening up opportunities for low and middle income country researchers, including those in sub-Saharan Africa, to own and lead research on priority projects that are important for their local communities. For example, this particular MinION, which is relatively affordable, is being used for field surveillance, as is shown in this image here, to detect and diagnose cassava viruses across farmers' fields within Africa, like we can see it being used here in Mama Mwangi's cassava farm in Kenya. I believe that for a sustainable and impactful outcome, we should be able to adopt, or we should adopt, a mixed model that includes working with partners to leverage technologies and expertise that will aid to develop and grow local capacities. I personally tend to think that if Bolog was alive today, he would advocate for the use of genomics to accelerate breeding for high-yielding, climate-resilient crops. I want to turn now to a capacity initiative that I mentioned, a community-focused capacity building initiative dubbed the Bioinformatics Community of Practice that was established in 2018. This initiative is a collaboration between scientists at the Becker Ilri Hub, scientists from the John Innes Center and Alham Institute in the UK, and later on ONT Technologies joined for the second cohort, as I will discuss. This initiative aims to train local young researchers from various sub-Saharan countries coming from different institutions, including universities and national research institutes. The initiative, unlike other short initiatives, is built along in-country residential training that shifts from short 1-2 week trainings to longer periods of training that expose the young trainees or the young researchers to a wealth of knowledge for a long period of time, where they can be able to grasp and put to practice what they learn. Being a residential training, it also encourages a lot of peer learning, And the trainees that have been trained eventually become the few who go on to train the many. And they continue to interact through a peer learning interactive platform. The bioinformatics community of practice training models revolve around three things mainly. The first one is to build and develop the knowledge and skills of the young researchers from various institutions in Africa through the delivery of modules, skills, and knowledge from experts around the world. Secondly is to empower them by demonstrating to them how to put to use the skills that they have learned through hands-on project-based learning. They develop the projects and they put to use the skills that they've been able to learn. And finally, the young scientists are deployed to amplify the impact of the knowledge and skills they have learned back to their institutions and countries, training their colleagues and other people that they can pass on the information to. And they continue to encourage each other through the community of practice, working together on various projects. So far, we've had 2 cohorts of the bioinformatics community of practice. The first one was in 2018 that I was a part of, which trained 14 fellows from 8 countries in an 8-month-long residential training on location here in Nairobi, Kenya. The training revolved around 7 modules covering various aspects of bioinformatics, including statistical analysis, quantitative analysis, training the trainer so that they can also be able to go back and be effective trainers. And it culminated in 3 mini projects, one of which we are going to focus on today later. These are the fellows that were trained from that first cohort. The second cohort trained 15 fellows from 9 countries. This was a residential training for 3 months here in Nairobi, Kenya again, and it focused on 3rd generation sequencing where ONT was a partner. They were trained on how to perform next generation sequencing using Oxford Mean Ion, and also to do the subsequent analysis for various applications. It also culminated in 3 mini projects that the fellows continue to work on interactively to date. And these are those fellows that were trained in the 2021 cohort. Where are some of those fellows right now? What have they been able to do with the knowledge that they acquired? Just a few examples to demonstrate. Ahadi Buhingane Birindwa from the University of Angelique in Congo recently won a UNESCO TWAS grant to open disease diagnostic lab within the university using ONT technology and subsequent analysis to study diseases within Congo. Beatus Limo, who is a lecturer from Nelson Mandela Agricultural Institute for Science and Technology, after his training, was seconded to the National Institute for Medical Research to carry out a project as a postdoc working on molecular surveillance of malaria parasite populations and anti-malarial drug resistance to support malaria control and elimination in Tanzania. I myself, with a master's degree in plant breeding and biotechnology, I received the training in bioinformatics, and now I work as a bioinformatician at ILRI, and I'm heavily involved in capacity building initiatives, not just in Kenya, but with Becker partners around Africa. And I was also lucky and privileged enough to be one of the trainers and facilitators for the second cohort of the bioinformatics community of practice. David Waweru from the second cohort of the community of practice currently works at the Institute for Primate Research in Nairobi, Kenya, where he is working to set up Oxford Nanopore facility to study toxins and antivenom development from snake bites within Kenya and the region. Idrissa Dieng from Senegal now works as part of a portable disease laboratory going through Senegal and its environs, studying zoonotic diseases, including Ebola and the SARS-CoV-2 group of viruses. Now I shift my attention to African-led genome sequencing of Lablab and African yam bean orphan crop genomes. So this project on sequencing orphan crop genomes is a culmination or something that was developed for the community of practice fellows from the first cohort to continue to work together and put together all the skills of bioinformatics that they learned from the 8-month residential training. Why did we choose to study these two legumes, Sphenostylis tenocappa and Lablab papyrus genome? They are quite important legumes for food, feed, and nutrition security, not just in Africa, but globally. The grains are very rich in protein and macro and micronutrients. The leaves are valuable sources of hay, silage, and forage. And the tubers of the African yam bean are high in protein, higher than some of the other tubers consumed, including cassava, Irish potato, and sweet potato as well. In addition, they are also adaptable to a range of climates, soils, and fix biological nitrogen in the soil, hence are able to improve soil health. I will start with genome sequencing of the Lablab papyrus legume. This was a project in collaboration with the Feed and Forage Development Team here at ILRI, led by Chris Jones. We wanted to develop a long-read genome sequence for this legume that is currently not yet published. There is an existing genome that was published using Illumina short reads by the African Orphan Crops Consortium in 2018, and we wanted to leverage what is already available and improve on that using third-generation long-read technology. So in terms of the Lablab genome sequencing, there's already an existing Illumina assembly, as I already said, that has been published, estimated to be about 423 megabase pairs. What we did was to generate 4.6 million Oxford nanopore reads with a mean read length of about 6.7 kb from R10 and R9 flow cells from the same cultivar that was used to generate the first Illumina short read assembly, that is cultivar Lablab 147D. From 28.4 gigabytes of data that provided us a 67x coverage of the estimated genome, we were able to use the long-read genome assembler called Fly to generate a base assembly that was then polished using the publicly available Illumina data to generate an assembly of 10.9 MB as an N50, a huge improvement from the previous N50 on the short-read assembly of only 697 KB. Looking at this radar plot, we can be able to see the significant improvement in terms of contiguity for the long read assembly in terms of the assembly size, the N50, as well as the largest contigs that we were able to observe. Leveraging the technology from Face Genomics, we used high throughput chromosome conformation capture, or HiC, from the US, from Face Genomics in the US, and we were able to cluster our chromosome into 11, or our contigs into 11 pseudomolecules covering 417 MB in genome size, which covers an estimated 98.6 of the estimated genome size. Assessing the quality of our genome using single copy orthologs using the BASCO tools, our genome had a 99% of the genes in the Embryophyta, Eudicots, and Fabalis databases, as we can see in this BASCO plot. With a high quality chromosome scale assembly, we could be able to then proceed to do gene annotation. We first sought out to do in-house genome annotation using the Fananotate pipeline to generate around 26,000 gene models. Then collaborating with an internationally accredited team in genome annotation that has even worked on the wheat genome. That's the plant genome and systems biology team from Helpmont's team in Munich. They also ran their own plant annot pipeline, and we worked together to generate a set of 30,922 models of which 24,972 of those we consider as protein coding genes, as they have no homology to any transposable elements. From the non-redundant set of 24,972 protein coding genes, we were able to recover 97.3% of the BASCO single copy orthologs, as well as 96 and 94% from the UDCODES and Fabalis databases, respectively. Our high quality genome enabled us to also do an in-depth look at the gene and repeat patterns on the Lablab genome, or on the Lablab chromosomes. In this Sakas plot, we confirm characteristics of the Lablab chromosome published in earlier studies. Together with the team from the University of Southampton and PGSB in Munich, we did an in-depth study of the repeats that cover about 43% of the Lablab genome. Long terminal retrotransposons, or LTR-RTs, were the most abundant of the transposable elements, and those transposable elements cover about 28.1% of the Lablab genome. Both the tandem repeats and the unclassified repeats were found to concentrate within a distinct overlapping cluster at the point peak density on each chromosome, indicating that these are likely to be centromeric repeats or centromeric regions. A karyotype study on Lablab papyrus suggested presence of metacentric, submetacentric, and subtelomeric chromosome structures. Looking at some of the patterns that we see in the Lablab chromosomes on this Sakas plot, on chromosome 1, we noticed the peak of genes and the repeats on opposite ends of the chromosome, on chromosome 1 and also on chromosome 8, suggesting that these two could be telometric chromosomes. We were also able to do an in-depth study of the gene families within our Lablab genome, comparing it to other closely related species. We did a comprehensive gene family analysis, comparing our Lablab genome gene families to its closely related species, including common bean, that is Phaseolus vulgaris, Adzuki bean, Vignia angularis, bark clover, Medicago traculata, and pigeon pea, that is Cajanus cajun. We found about 14,000 of those genes being unique to Lablab. We also did an analysis for the expanded and contracted gene families, and among the expanded gene families, a functional enrichment of those revealed that we have genes expanded associated with oxidative reduction, catabolic and carbohydrate processes, and even drug metabolic processes. With our high quality and chromosome scale genome, we were also able to study the domestication history of Lablab, which we did in collaboration with Dr. Mark Chapman from the University of Southampton. Several 2-seeded and 4-seeded Lablab accessions were resequenced and mapped to our Lablab assembly with a mapping rate of 95% or higher. Data was also gathered from other outgroup species available to generate a set of 67,000 SNPs that were used to generate a phylogenetic tree to study the domestication of Lablab. The cladogram here confirms previous work suggested 2 domestication events in the Lablab evolution, separately occurring for the 4-seeded and the 2-seeded accessions of Lablab. We could also utilize our high chromosome scale genome assembly to study the genetic diversity of Lablab. Working together with the ILRI Gene Bank team in Addis, a set of more than 1,800 individuals from 166 accessions were genotyped using DATSEC technology to develop SNPs, which were filtered to enable us to do an in-depth diversity study. The studies we did were able to cluster the accessions into 4 main clusters. The first cluster here contained mainly accessions from India. The second one contained accessions from Kenya and India. And the third and fourth accessions contained, the third and fourth accessions contained, clusters contained accessions from the East African region. So with what we were able to learn from the first genome sequencing project that I've just talked about in terms of Lablab, we are also implementing the learnings that we've done from that sequencing project to further sequence another genome that is from the African yam bean. Currently, there is no documented genome sequence for the African yam bean. And just to remind you that this legume produces edible pods and tubers. So it would be very interesting to understand the underlying genetics, which is not possible without genome sequence. So our goal is to generate an African-led chromosome scale genome assembly to aid in crop improvement for genetic gains of this crop in Africa. We estimated the genome size of AYB to be somewhere between 800 and 838 megabase pairs using flow cytometry. And we were able to sequence short reads and nanopore reads from right here at ILRI at the sequencing facility at Becker. We generated about 103 million reads from the Illumina sequencing platform and about 6.7 million reads from the nanopore platform. The nanopore platform gave us a coverage of about 40x of the estimated genome size. Having been able to generate both the Illumina and nanopore reads, we were able to test various algorithms for de novo genome assembly. We tested Flyer as a tool and Redbean as a tool for long read de novo assembly, as well as Shasta, and we could see the effect, or we could see the various genome statistics that we found from the various algorithms. We also went ahead to see the completeness that we achieved from the various algorithms. So testing an ONT only assembly, testing an assembly that was a hybrid using spades from ONT and Illumina, and also looking at the effect of using different base callers algorithm or base calling algorithms from ONT data. Using high accuracy or base calling algorithms gave us a pretty good assembly. And then what we observed is that when you're able to use a base assembly developed from ONT and then use the Illumina reads to polish it, we could generate a relatively high assembly where we were able to recover about 98.1% of the single copy orthologs through the BASCOL tool from the EmbryoFita database. We've just recently received our data back from Face Genomics, and we believe that we now have a chromosome scale assembly from high throughput chromosome conformation capture, or HiC. We have clustered our contigs into 11 pseudomolecules that we believe will enable us to publish a chromosome scale assembly with a massive improvement in N50 to about 69.5 MB. This current assembly from HiC has also a relatively high score in terms of BAS scores. We've been able to recover 98.5% of the single copy orthologs from the EmbryoFita genome and also from the Eudicots and Fabalis genomes. So we intend to carry on and push through to generate a genome annotation for the African yam bean, as well as try to understand the genes that are involved to enable the African yam bean produce edible pods and edible tubers. This is going to be a very interesting study, and we have a lot of interest from scientists within the African continent. So with this journey of the Bioinformatics Community of Practice Initiative and the Lablab sequencing projects and the AYB sequencing projects, I'd like to share a few lessons that we have learned along the way. What worked for us? The longer residential training model worked very well. The fellows had enough time to grasp new skills, put them to practice with the hands-on practice projects, and they also had an accelerated rate of learning through the peer interaction, where they were able to stay together here in the long residential training periods. Collaborations were also key in making sure that the trainees got the best experts to teach, train, and mentor them. Through a North-South collaborative initiative, working with scientists from UK, from Germany, we have been able to generate a high-quality Lablab genome assembly and gene annotation that is already proving useful for researchers not just in Africa, but also beyond. Challenges are growing, are part of any growing process, and we also had our fair share of them. Key among those challenges was funding. Among the reasons why the second cohort of training was short, it was only 3 months as opposed to 8 months for the first one, was because we had some budget cuts and some of the PIs had to come in and support the budget. To complete the objectives of the sequencing projects, various programs have also contributed to see it through to where it is today. Keeping the fellows engaged after they leave the residential training is also one of the other challenges. Some have gone on to take advantage of opportunities that have come up traveling outside of Africa. However, I think what we have really noticed and what we want to really advocate for is to provide opportunities where young Africans can be able to put to practice the skills they learn from initiatives within Africa, and also for those who have been trained outside to have the motivation to come back and put to practice those skills to push through the priorities that are important for their local countries and local communities. Initiatives like the African Orphan Crops Consortium, the Bioinformatics Community of Practice that I've talked about, the African Biogenome Project are great initiatives that need support and funding from key donors to ensure that they are able to create an environment for genomic research that will solve Africa's key issues. Moving forward, we have learned a lot from our journey, and we want to put to practice what we have learned with the Lablab genome sequencing project to push through the African yam bean project to see it to completion and publish an African-led chromosome scale assembly, and also to push that and learn and what we've learned to be able to complete other genomes that are in the pipeline. There's one genome that is in the pipeline from the second cohort of bioinformatics from the Dovialis Kafra orphan crop, and be able to generate more resources for not just the African content, but also for the world to take advantage of. We will also continue to seek more funding to train more scientists within Africa to create a vibrant research community within Sub-Saharan Africa. A few take-home messages, very simple. Despite the challenges that we face, a rapidly growing population, resource constraints in terms of capacity and funding to build a sustainable agricultural productive environment, we have a wealth of crop diversity that are highly resilient and nutritious, and we can scale them up to meet the demand through use of technologies like genomics, and with the right partners who share our vision and can continue to provide opportunities to learn and develop the required capacity of knowledge for local researchers. We will be at the forefront of generating resources like high quality chromosome scale plant genomes to support crop improvement for food, feed, and nutrition security in Africa and beyond, taking full advantage of the bounties of the continent's biodiversity. I would like to appreciate and acknowledge everybody who has been involved in this journey. All the fellows from the 2018 cohort, the first cohort, have been heavily involved in analysis and taking part in the Lablab and African yam bean whole sequencing, genome sequencing projects. And also our wealth of collaborators and mentors from here at ILRI, from the UK, from Germany, scientists in Nigeria as well, from from Addis, sorry, and also all the people who've helped us within ILRI and beyond in terms of technical assistance, and all the funding bodies that have been involved to bring all this work and this journey to fruition and to see it through. Thank you very much, and I will be happy to take your questions.